Alright, today we're talking about passive transport. Passive transport, when we're talking about transport, I want you to still think about the cell membrane because we're going to be talking about transporting molecules or macromolecules in or out of the cell. And in order to do that, we of course have to transverse the cell membrane. Now, passive transport, we distinguish using the word passive because passive transport is going to be the specific kind that does not require energy. Now this is going to only occur when there's a concentration gradient. So we'll go more into that word concentration gradient in a second. But you just always remember movement is from high concentration to low concentration. There's three main types that we're going to talk about in passive transport. Diffusion, which is just a molecule, right? That's small enough to go through the cell membrane is going to diffuse into or outside of the cell. Remember, from low to high concentration. So here you have a high amount or a high concentration of whatever particle this is on the outside of the cell, and it's going to want to move to the inside of the cell where there's a lower concentration. The second type is going to be osmosis, which is when water is going to move from high to low concentration. And then the third type is going to be facilitated diffusion. Now this is still passive transport, but it occurs for molecules that are going to be too big or for some reason can't pass through this cell membrane. So what we have here is going to be an integral protein, right? Or a transmembrane protein that's going to form a channel that will allow for the free, with no energy costing, transport from high concentration to low concentration of these molecules, right? And that's what we call facilitated diffusion. It's going to be it's still diffusion, right? These particles are going from high concentration to low concentration, but it's facilitated by this protein channel. So you have here, right, before and after. This is what the concentration gradient is. In this one rectangle, you have 6 x's, and in this rectangle, you have 2 x's, right? Now imagine I have a barrier here. When I release this barrier, the molecules are going to want to move from side B, right, to side. So they're going to move, right? They're going to move to side B. That way you have an equal amount on each side, right? That's just how molecules exist. They don't want to be around too many, so they're going to go to where they're equally spread out. And that's what we call a concentration gradient, from high to low until they're equal. So here's another way to look at concentration. You have, if I had a, let's say you have a big glass of something, and if you ever notice your glass is really, really dark at the bottom and maybe really light at the top, you've probably instinctively already just shook up that glass, right? And what you're doing is creating an equilibrium of concentration, right? So this is going to be equally distributed. This is going to be concentrated at one side and dilute on the other. So that's what we mean when there's a high concentration to a low concentration. So the selectively permeable membrane. So that's going to be, when we say that, right, we're talking about the cell membrane. The cell membrane is selectively permeable. So that means some things can get through the cell membrane, but not all things. So usually only good things will be allowed to pass through. Whether that's calcium or whatever it is, those molecules will be allowed to pass through. And when they are allowed to pass through, then they can undergo passive diffusion. So you have the lipid cell membrane, you have a bunch of things outside in the extracellular space, right? A high concentration of this particle. Over time, these particles are going to seep through the cell membrane, they're going to diffuse through the cell membrane until there's an equal concentration on both the inside and the outside of the cell membrane. Just like in this water glass, if you had a semi-permeable membrane, the sugar, right, the sugar is not going to be able to pass through this membrane right here, but let's say the water molecules can. So instead of the particles moving, the sugar particles, which are too big to move through, the water through osmosis will actually move through and try to equal the concentration, so that the concentration, the ratio of water to pellets, is going to be the same on both sides. And you can see here it's actually working against gravity through the forces of osmosis. Now here's just a depiction of facilitated diffusion. you have these big particles outside the cell, and once these particles are obviously going to be too big to fit through the cell membrane, but let's say they come into contact with this membrane channel here, they will be able to just freely flow inside the cell because they're going to be the correct size. So that could be for glucose. So maybe the cell is always looking for glucose. It's always having to make its own. So if it ever runs into any glucose outside the cell, it wants to make sure it can get the glucose in. So maybe this is a one-way facilitated diffusion channel. So here are just some terms that we should know. You have a solute, which is going to be the part of the solution that is the dissolved, right? So in salt water, the solute is going to be salt. Then you have the solvent, which is the part of the solution that does the dissolving. So in the example in salt water, it would be the water. So the solute is going to be the thing that's usually was once solid and then was dissolved, and the solvent is going to be the water. The dots are the solute, and the solvent is the white space. So the water is dissolving the dots. So these dots represent each one particle, right? One particle of solute, and then this whole thing is going to be covered in solvent. A hypertonic solution is going to be a solution that has a higher concentration than another solution. So this is going to be water out, and the cell will shrink. So when the solution is more concentrated than the cell, right? So let's imagine that this is one cell and it's placed in a hypertonic solution. When the word hypertonic means that the solution is more concentrated than the inside of the cell. So let's say that these particles cannot diffuse out of the cell, water has to move. That means that osmosis will drive the water out of the cell in order to get the ratio of particles to water equal inside and out of the cell. So that will cause this cell to shrink. Right? So water is going to leave the cell. Hypotonic is when you have a solution that's lower concentration than another solution. So again, look at this picture. You see the dots here are more concentrated inside the cell. The dots here are less concentrated outside in the hypotonic solution. That means the water is going to enter and the cell is going to grow. So you have the water entering here because, again, they want to equal this ratio of particles to water, of solute to solvent. Right? And these are going to be really critical ideas, and they're not really intuitive. You're definitely going to have to memorize what this word means, because on a test, you're not going to have these diagrams. You're just going to have this word, and you're going to have to know what it means and what it results in. All right? And then you have an isotonic solution, right? So these dots are all equally spaced out in this whole picture. The ratio is going to be the same inside and outside of the cell. So the solution is of equal concentration to another solution. This cell will have water move in and it'll move out at the same rate. So this cell is going to stay the same shape. So this cell is pretty happy, right? Now what happens if a cell loses too much water or bursts from getting too much water? We call cytolysis, right? Cytolysis is the bursting of an animal cell when it's placed in a hypotonic solution. Now a hypotonic solution, we should remember, is when the water is going to go into the cell because the cell is more concentrated than the outside. So this is cytolysis when the cell bursts, right? The water is filling up the cell too much, so much so that it actually bursts. Then you have plasmolysis, which is when the shrinking of a plant cell happens when it's placed in a hypotonic solution. So plasmolysis, right? You have this cell wall that's not really going to change shape. But because the solution on the outside is so much less concentrated, the water is going to leave and then cause this inside cell membrane to shrink. And then finally you have turgid pressure, right? Turgid pressure. So that's the force that pushes out of a plant cell wall when it's placed into a hypotonic solution. So a hypotonic solution is going to be water coming in again, remember that? And then the water is going to be coming in at such a rate that it's going to actually push against the cell wall. And this is the normal state of a plant, right? If it's turgid, that's the normal state. The plant wants to be full of water. It's only a problem if this plant is filled with too much water, so much so that the cell walls actually burst. So here we have an example of a red blood cell, right? So a red blood cell placed in an isotonic solution, right? So it's as concentrated as the inside as it is outside. Those are going to be normal, happy cells. Nothing going on here. If this is a plant cell, the plant cell is going to be pretty happy. Then you have a hypotonic solution. In a hypotonic solution, you put cells in there. The water is going to enter, right, because the cells are more concentrated. The water is going to enter and actually burst some of these red blood cells. If you have a plant cell, hopefully that cell wall is going to be strong enough to prevent that from bursting. Then you have a hypertonic solution. And then these cells are actually going to lyse because they're so... It would be if you placed a red blood cell in salt water. The salt outside would be more concentrated than the inside, so the water would want to leave. And then you have these kind of shriveled up red blood cells. In a plant cell, because the cell wall doesn't really lose its structure, only the inside of the cell will shrink. And finally, just some more terms. You have diffusion, right? This is something we've already talked about. So that's the movement of molecules, right? The dots. That's when the dots actually move from high to low concentration. So if this circle is a cell, the dots are going to be the molecules, and like I said, the white space is just going to be the water. So in this situation, it appears that the dots on the inside of the cell are more concentrated than the outside, so the dots are going to move out. The particles are actually going to move out of the cell. The movement of water we refer to as osmosis. And osmosis acts the same way as diffusion, except we talk about water. Now water is going to actually cause the cell itself, if the water is moving out, the cell is going to get smaller. If the water is moving in, the cell is going to get bigger. Since there's more dots on the inside of this cell compared to the outside, the water is actually going to move in because it wants to get the ratio of water to dots equal. So that's going to cause this cell to swell. Then you have facilitated diffusion, which is going to be the movement of those molecules from high to low concentration, but it's going to be only possible with the help of a protein channel, right? So just like that very first picture we looked at, it's going to be maybe some kind of protein gate here that's going to allow these dots to leave the cell, right? So here's the protein channel. Because there's more dots on the inside, these dots are going to leave the cell. Alright, so this is just going to be practice. We're going to address this in class. Watch the video if you missed anything. I know it was a lot of stuff. This unit is definitely going to be tougher than our other ones, so make sure that you understand everything and take any questions you have with you to class.